The Raman effect clarifies the inelastic scattering. Chemical engineer and an alumnus of MIT in the US and IIT Bombay, distinguished alumnus as well. Dr. Premnath.
Well, thank you. Ashish, I'm stepping in for Ashish today, and Ashish typically has a nice essay to start some of these events, and that's something which I'll try and do in small measure. We have very limited time, so we'll try and do that as well. Of course, as Anu just mentioned, today is the National Science Day. And every year, as she mentioned, 28th February is celebrated as a National Science Day to mark the Raman effect. His classic paper, Raman's classic paper with K.S. Krishnan, titled The New Type of Secondary Radiation, was actually published in Nature on 31st March 1928. And the announcement that he made was February 28, 1928. So I always wondered how the process worked in those days when we didn't have email, and how people managed to communicate papers around that time as well. By the way, we were blessed to have Sir C.V. Raman's presence at NCL on 3rd January 1950, when NCL was formally opened by Pandit Nehru. And this institution has actually had a long relationship with Sir C.V. Raman right from its inception. Sometimes full of bitter disagreements and science policy debates, but nonetheless, I think something very characteristic of all of us as scientists, to argue, to debate, and I think any day it's better to argue than to be indifferent and disinterested. So that's something which I want to keep alive today as well. I want to tell you a little bit about I want to excerpt a few things from one of Dr. Raman's talks today, because he represents a different point of view, and it's important to keep in mind. As Anu also mentioned, this year the government has announced the National Science Day theme as Indigenous Technologies for Viksit Bharat. And as all of you know, I have chosen to do whatever I do in directing science towards socio-economic development and looking at future industries for the country and the world. That said, today's day belongs to Sir C.V. Raman, right? And the spirit of science as he saw it. Okay, so I'll keep it aligned with that. On 22nd December 1968, Sir C.V. Raman delivered a lecture on the foundation stone laying ceremony of the Community Science Center in Ahmedabad That was probably when Dr. Sarabhai was there, titled "Why is the Sky Blue?" And in that talk, while the title says "Why is the Sky Blue?", it was actually a discourse on the spirit of science. And there are several nuggets of wisdom for the young scientists in this audience. And I want to share a few quotes from that lecture. The first one is a quote on nature as an inspiration for science and its sense of wonderment. We often think that somehow we are losing the sense of wonderment in trying to pursue certain output-driven activities. So it's important in that context. And I quote, "You learn science by keeping your eyes and ears open and looking around at this world. The real inspiration of science, at least to me, has been essentially the love of nature. Really, in this world, wherever we see all kinds of miracles happening in nature. To me, everything I see is something incredible, something absolutely incredible. We take it all for granted, but I think the essence of the scientific spirit is to look behind and beyond and to realize what a wonderful world it is that we live in. And everything that we see presents to us not a subject of curiosity, but a challenge to the spirit of man to try to understand something of this vast mystery that surrounds us. Science continuously attempts to meet this challenge to the spirit of man." He's linking science to the spirit of man. He later goes on to reiterate, "It's not necessary to hunt around the textbooks to find problems of science. You keep your eyes open and you see all around you. The whole world bristles with the problems of science, but you must have the wit to solve it. You must have the strength of mind to keep going at it until you get something. This is the lesson which I want to bring home to the younger generation in front of me." There's another quote which is on the process of scientific inquiry. He says, "Don't read any book about it. Don't ask your teacher. Let us sit down and try to think out this problem. Why is the sky blue? Look at it as if it's a completely new scientific problem about which nobody has troubled himself before. You sit down and think it out, and you will find it the most exciting thing to ask yourself that question and see if you can discover the answer for yourself. Now I'll put it to you in this way. The best way to answer a question is to ask another," right? And he illustrates this through his lecture, and I recommend many of you young people to read it. There's also another quote on the endless quest that is science. He says, "What is science, and how can we in this country hope to advance science? How can we try to make ourselves worthy of our ancestors in the past?" That is the real topic of my lecture. Science never stops. It's going on. The more you find, the more appears, and that you have to find. That's the attraction of science, provided you're not too distressed too much by other people getting in front of you. Don't bother about them. The real point is that it is an endless quest, and every new discovery opens new paths for discovery. New questions arise, requiring new answers. I think this is something which, again, we lose track of trying to forget that this is an endless quest where one problem leads to another. There's no really end to it. And so we are just participating in a journey. And sometimes in our own activities, we tend to box it into endpoints, which may not be so at all. The final quote is on the utility of science. And I'm sure some of my colleagues would find joy in listening to it. "I want to stress the philosophy of my life, never to ask what is the use of all this. And I told you before, it is the striving that is worthwhile. Because we have certain inherent powers given to us to use, observation and thinking, we must use this. The more we use them, the sharper they become, the more powerful they become, and ultimately something will come out of it so that humanity is benefited, science is benefited. Ultimately, the aim of scientific knowledge is to benefit human life." He further goes on to say, "The most important, the most fundamental investigations, though at first might seem an abstraction of nature, are precisely those which in due course affect human life and human activities most profoundly. This is a very heartening thing because one should not think that scientific work, in order to be valuable, should be, I add immediately, useful. Scientific work is valuable because it will ultimately prove its value for the whole of human life and human activity. And that's the history of modern science. Science has altered the complexion of things around us. And precisely those scientists who have labored not with the aim of producing this or that, but who have worked with the sole desire to advance knowledge, and not necessarily to run a rat race, right? I'm adding that, ultimately prove to be the greatest benefactor of humanity. I think there are a lot of lessons to be learned from Dr. C.V. Raman's speeches, talks, and his life. And many young people may want to go back. There are a lot of free resources available online. Many of these things have seen, you will see recent examples as well. If you look at the recent Nobel Prizes, especially, for example, if you look at CRISPR or the modification of the mRNA, you will find what he's saying has a lot of meaning. But that said, somebody has to look at uses as well. I'm not trying to say that that's not important. I'm just saying that you need to advance knowledge as well. So dear young friends, you may wish to imbibe and live the spirit of science that Sir C.V. Raman spoke about. One of a sense of wonderment of nature, a spirit of scientific inquiry, the desire to know and discover knowledge, cultivate the sense of continuously rising up to challenges posed by nature, and to contribute to this collective, valuable activity of humanity, which we call science. While learning from the wisdom of Sir C.V. Raman, let's also dedicate ourselves to the mission of this lab, as christened outside, to advance knowledge and to apply it for the good of the people. With these words, I welcome you all to the National Day celebrations at NCL. I welcome Professor Ruchi Anand to NCL. And I'm grateful she has taken the time to join us here today on this occasion. We look forward to your talk. Yeah, thank you.
Thank you, Premnath, for bringing us back to understanding why we are all here and why we struggle every day. So now I invite Professor Ruchi Anand to deliver the Distinguished National Science Day lecture. Professor Ruchi Anand is currently the Institute Chair Professor, Department of Chemistry at IIT Bombay. Ruchi completed her PhD in 2004 from the Department of Chemistry and Chemical Biology at Cornell University, US. After conducting postdoctoral research at Sloan Kettering and the University of Pennsylvania, she joined IIT Bombay in 2008 as an assistant professor. Her laboratory employs a combination of X-ray crystallography, cryo-electron microscopy, and now she has installed one of the biggest facilities around here. So any students want to use this facility, collaborate with her, please feel free. She also has developed biochemical and biophysical tools to understand molecular mechanisms with implications to human health. Her current research interests follow a multi-pronged approach where she has contributed to two major areas, antimicrobial resistance and biosensors development. Towards the first goal of combating drug resistance, she focuses both on unearthing enzyme systems that can serve as new therapeutic targets, as well as is involved in understanding the origins of antibiotic resistance itself. Her recent work with bacterial transcription factors has paved way to structure-guided development of biosensors for aromatic pollutants. She has a list of long accolades. She's serving as on the editorial advisory board for many ACS journals, JBC, Asian JOC, JACS, and now she's also the Associate Editor of Biochemistry. She's the recipient of National Academy of Sciences Fellowship, National Women Bioscientists Award recipient from DBT. She has won the CRSI Bronze Medal for 2020. She's the recipient of the DBT Wellcome Trust Alliance Senior Fellowship. She's also been admitted to the Indian Academy of Science in 2022 and inducted as an Indian National Science Academy Fellow in 2023. And then we just found out that she's also one of the 3 recipients of the Sun Pharma Science Foundation Research Fellowship of 2023. In addition to all her achievements, I happen to know that she gives great talks. So with this, I request Ruchi to deliver her lecture on sensing platforms for aromatic pollution detection.
Thank you.
It's a pleasure to be here, and I thank Anu and Sarika and the organizers for inviting me here today. And I'm really glad to be part of this program, and I hope I'm able to bring across the science we do in a simple fashion. And I'll just get my mic. I think, good. Yeah, so thank you so much for this invitation, and I'm very glad to be here. And I'm going to talk about sensing mostly today, but I thought that just to start, let me tell you that our lab is like two different divisions that we work in, but all the work we do is got to do with proteins, enzyme mechanism, and trying to understand and use them to application-based approaches. And we use a lot of techniques. We collaborate a lot to be able to answer two problems that we are working on. One is trying to develop biosensors for hard to detect pollutants like benzene, toluene, phenol, etc. And we start with nature's approach there. And I'm going to talk mostly about our work on this side today. The other approach is got to do with using same enzymes, but we try to understand why bugs who were not at all resistant, they suddenly become resistant, and what has happened to them, and what mechanisms that they have gotten to be actually reach there. So in this regard, what we focus a lot on is actually, I don't know whether I'll be able to fast forward this movie a little because it's a very long one, and all I wanted to show you was this part, that most of the drugs that go and bind, you know, erythromycin and lincolnomide, streptogramins that you know, many of the drugs that you know, they bind to this particular machine. This machine makes peptide bonds in our body. So all proteins in our body is made by this machine called the ribosome. And as the ribosome functions, you can see that the protein comes out. So RNA is here, something happens inside and it comes out. And many of the drugs do not allow this protein to come out. And those are the drugs called the macrolide class. So these are all the drugs which basically what they do is they stop protein synthesis. If we cannot make proteins in our cell, all the catalysis that happens is using the proteins will stop. So these drugs are the ones, and bacteria become resistant to these drugs, this class of about 50 to 60 antibiotics, and we try to understand. And how do they do it? I showed you, right, there was this protein coming out. If it sits in that protein tunnel, then no longer the protein is able to come out. But what the bacteria do is they develop small modifications. It's like not allowing that antibiotic to bind. And they have enzymes like erythromycin-resistant methyltransferase, which actually put small markers, methyl groups. And once that happens, these drugs are unable to bind and resistance develops. So in our laboratory, a lot of work we are doing in trying to understand if we can develop inhibitors or modified antibiotics. And these are some of the past students that have worked on it. And this is recently that we collaborate with synthetic chemists, Swarn Kulkarni at IIT Bombay, where we modify these macrolides, solve the structure of these macrolides in using cryo-electron microscopy now. And then we are able to see the drug in the pocket and see how we can modify it. Basically, what we are trying to do is circumvent resistance, come up with either mechanisms such that old antibiotic starts working or tweak the new antibiotics slightly so that we can make them work. But today I'm not going to talk about it. I'm going to talk about this is just, I wanted to give you a flavor of what all other things we do in our lab. Today I'm going to focus on this problem on 90% of the time that I work, is what I came with the thought of doing. And here we started with an approach where we started with just trying to understand sensing, and then we have gone on to be able to do work which has got to do with device development in collaboration with groups at IIT Bombay. So let me, I don't even have to say too many words that water, having safe drinking water is a very important thing. And most of the pollutants, and if you know, every house has an aquaguard, right? Even every place you go, you don't drink water, at least in several parts of India, where you can just take it out of the tap. Because you want some kind of purifications to some heavy metals, some ion exchange, some kind of columns are put in there, and then you check for pollutants. Natural, some small number of pollutants are actually, or essential pollutants are kind of removed by that process. But the problem is pollutants such as benzene, like which can happen due to an oil spill, or other pollutants which are like toluene, hydrocarbons, even PCBs that we talk about, they don't have any functional groups that are so easy to detect. And when you have difficult to detect molecules, then what happens is that they remain undetected. So you may have it in your water, but you may think, I don't have it, because you never have any detection systems to actually detect them. So that's a problem, that is a growing problem, because many kinds of materials have these kind of compounds in there, and over time they enter, and they're very stable. They enter the cycle, and then they're around in groundwater and many other sources. So we thought that why don't we tackle this problem, at least with respect to aromatic pollutants. Up till now, they are just called as organics. People actually don't know exactly what is in that organic portion. So we are only targeting part of the organic, which is aromatic pollutants. So what we decided was, there are many approaches. People actually develop different kind of sensors for capturing these small molecules, but the problem is there is always some kind of not so, you get a lot of molecules that come your way, and you're not able to make very specific, stable sensors. Now, so we thought, why don't we take help from the nature? So if you go to nature at certain places, what nature has done is, it has been able to develop mechanisms, like there are bacteria who have naturally developed, for example, in phenol contaminant. So dye industry and all these areas have a lot of phenol contamination. What they have done is, they have developed natural mechanisms where what happens is, they generally use glucose as their main source, but what they have developed is they can sense, for example, phenol. And so they have sensing systems which they're sensor proteins which will sense the phenol levels, for example. This is an example. And then they will turn on genes which will eat phenol. So if you are under a lot of stress, if the bacteria is under a lot of stress, instead of eating normal things, it starts eating phenol. So that's a good thing if you think about it. Now, not only is the bacteria sensing the pollutant, it's also getting rid of it. So in our case, we said, obviously you can always go the next step where you can try to also develop systems to get rid of it, but we first thought, let's take this natural sensor and see if we can translate it outside the system, and can we then perhaps tweak it further. That was the ethos with which we started. So this kind of systems where you have these bacteria which have sensors which turn on in presence of a small molecule, and then they basically they kind of eat them up, and then you kind of get rid of them. And these are the class of molecules we thought first we will start with. So how does this work, right? What I'll do is, I'll take out the sensor protein from the bacteria, and this is the sensor protein. And this work was done by Chamita, who's long left our lab and is in Harvard currently. But what we did, what Chamita started doing was, we tried to understand how these sensor systems work inside. So instead of telling the whole machinery, I will just tell you, what we did was, every time you want a sensor, you want a place where the molecule is detected. Then you need some kind of a signal so that you can actually read the detection. Right? I'm able to bind something, but I need to read it. So this protein is perfect in that sense, that this is the place, this box, let's put it this way. This box is the place where phenol goes and bind. Hopefully this box will become a protein very soon, as I'll show you. And in the middle box, the minute phenol binds, there is an action where it then starts producing ATP. So what is happening here is, this is the protein. I'm only taking these boxes. I'm neglecting this box right now. This is the blue part of the protein where phenol will go and bind. Once it binds, there is some kind of a change in the system. And as a result, what happens is ATP starts getting hydrolyzed. ATP starts becoming ADP and phosphate, and we can detect this reaction and by a colorimetric assay. And what then we can do is, we can use this as a sensing assay to sense against other pollutants. And what we see is, because it's an evolutionarily conserved system and already the bacteria has seen all kinds of pollutant systems, has chosen phenol as its target, what happens in this case is, it is able to, with high fidelity, only select phenol, right? So what we have done here is good and bad. So what is the, let's say what is good about it, it's very selective. What's bad about it, I can only do one pollutant, right? So I'm stuck with not being able to do a repertoire of pollutants which is present in my water. So I have a water, it may not have phenol, it may have benzene, it may have toluene, it may have something varied compounds, but I'm pretty much stuck. So there are good points and bad points to the system that we are able to do. But before worrying about good or bad points, we said, is it even translatable? Can we go ahead with the system? Because most of the thoughts about proteins, at least with most people, is I touch it and it's going to fall. You know, oh my God, this is a protein, you know, it's not going to survive anything. So what we did was, we wanted to make sure we already have a selective system. We want to tweak the sensitivity, and over the talk I'll tell you how we are trying to change systems to improve sensitivity. Whether required or not depends on what application you are doing, right? And we also want it to be cost effective and reproducible in a stable sensor. So what we did was, we looked at the protein, and one of the ways in which you can actually make a protein really, really effective is by making it stable, so that you don't have to put it at 4 degrees, you don't have to look at it and it just like, you know, disappears. This thought should not stay in the mind of the person who's trying to develop a sensor. So, so what we did was, we tweaked the protein. That means we tried to figure out what is the best way to actually have the minimal domain such that it's functional. You're able to get sensing and ATPase activity both, but it is not that sensitive to degradation. And in doing so, we tweaked the boundaries of the protein in such a way that now this protein is actually able to function at around 70 degrees centigrade, is okay for many, many weeks outside, and reproducibly is able to give activity at different temperatures. Up till about 60 degrees, we are able to get very good activity. Activity means it will have phenol, and it will sense phenol, and it is then going to hydrolyze ATP. Because sensing is not enough, you need something which will tell how much sensing, what's going on. Some readout is there. And so in this case, the ATP hydrolysis, we are turning into a green color. Right? So we need sensing and readout. Both is happening in this one system. And not only that, we tried to, after that, add lots of other pollutants and went down to about 10 ppb using this system. So what this is telling you is that this is a good sensor, because obviously it has been evolutionarily found. Most of the sensors of this class were actually found to be very unstable outside the cells, but we were able to engineer this protein so that it's really stable. So one winning point is stability, long time, is very selective, can go to low concentrations even in a milieu of pollutants, and works actually on real samples. So this is two sites, but we have now done it in many sites, about 10, 15 different sites we have tested this particular sensor. So like I told you, every time when I was talking about our work, I was saying that there's a good part and a bad part. So we wanted to address, so some good parts we get lucky, and we had a system which is working, but that's not where we wanted to be. We wanted to expand the repertoire, have many other pollutants, for example. So I will give you an example of one engineering and design that we did, and then there were many other engineering and designs that we did. But to do that, we needed to know how this protein senses phenol. You know, how does it know, how is the phenol entering, what is going on? So I will give a snippet of this. What we did was, the easiest thing to do now is to determine the structure of this sensing domain. So Shamita in a lab a couple of years back quite a lot of years back, determined the structure of this sensor protein. And what she showed was that this phenol sensor, it's a phenol sensor, it binds, is anchored by a tryptophan and a histidine. So an indole ring and an imidazolian ring is the one, the nitrogens of those actually OH hydrogen bonding. So there is like having some kind of a hold on the top, and the rest of the phenol has a lot of hydrophobic amino acids. So it's a hydrophobic cavity. This is hydrophobic with an anchor which is partially hydrophilic, right? So you have the phenol, you need to anchor the OH group, and then you need the rest to be able to have a nice pocket, and it's a very snug pocket, right? And so what this three-dimensional structure helped us to is to understand the pocket really well. So once you have the pocket and you understand the pocket, the next step is, it's only a phenol sensor. Can I now look at the pocket and come up with a new design of a new different sensor? Otherwise, I'm limited in my approach, right? So, so the thing that we thought was, can we, for example, make it into a benzene sensor? So what do you need for a benzene sensor? You need this hydroxyl, the position where the hydroxyl is to be satisfied such that the hydrogen bonding goes away there. They're not looking for hydrogen bonding anymore. And what else you need is, the rest is just the hydrophobic part of the pocket will take. Now you look at it, it's phenol. We don't want the hydroxyl part as a kind of a problem. And then what we want is it to be able to accept the benzene molecule and not accept the phenol. We don't want a promiscuous sensor either, because then we don't know what we are sensing again and how much of what is there. So what we did is we looked at this sensor design very carefully, and what we were aiming at was develop sensors for hydrocarbon, this aromatic hydrocarbon class. So there is actually no detectable functional groups. OH, you can do stuff and actually detect. This is actually a bigger problem that we were addressing than phenol detection, right? So what we did here was, we thought a lot, I would say a lot, and what we did was we came up with this design, okay? What we came up with the design was, you see there's a histidine and there is, you can have there is a imidazolian nitrogen over here, which is hydrogen bonding, and what we did was we converted that into a tyrosine which has a benzene ring, a phenolic group. So we have the phenol embedded inside the protein now. So what we are doing here is, we are not disturbing the enzyme's happiness. All we are doing is we are putting an OH group where the OH group is supposed to be, except now the phenolic OH group cannot be there because there's a steric clash, right? So now what we have done here is, we have created a system where another OH has replaced the OH of the phenol. Okay, and so now you have a good place for benzene, and it works. It is not any kind of magic here. It works really well as a benzene sensor. Doing this was the first approach where we are able to convert into benzene, and it would take toluene to some degree also. But then we were able to engineer the rest of the hydrophobic pocket and later develop sensors for all of these, depending on how big we want it to go, up to a certain limit. So it's not like, it's magic that you can keep thinking and doing. At some point, our thought fails us and nature wins against us. So we can discuss it. If you want to ask me, I will tell you the problems. But what this did for us was, it was able to help us make many, many sensors, and we could, at our own ablation, come up with at least some modifications and come up with sensors of hydrocarbon monoaromatic so far, and change the groups depending on the need at any time. So we even also collaborated with Shubhankar was the graduate student who just graduated actually, and he actually was the one who spearheaded this work. And what he did was, he collaborated with a group in IIT Bombay itself, Rajdeep Bandyopadhyay, and what he did along with his student was, they came up with a device which is very cheap, an LED based device, and it has 4 different channels, and in these channels you can then benchmark each of the pollutants, and you can have a system where you can actually then make your 4 different pollutants together. So what we are doing is individually, there is a different sensor. Remember, we made these sensors. One is a phenol sensor we have made, one is a benzene sensor, one is a ethyl benzene sensor, and so on. We have made xylanol sensors. So depending on the pollutions, we have 4 different sensors, and you can have 4 different channels where you can use this particular device at. So that was good. Shubhankar got a little bit happy that he was able to do, but our aim was not to stop there. Our aim was to miniaturize this device further and try to see if we can do immobilization of the protein and try to see if we can then make something like a glucose sensor. If you remember, you have this diabetes glucose sensor, and it is completely protein based. Right? And it's a very successful diabetes sensor. Everybody uses it. It's glucose oxidase is what is used in it. So we thought, can we also go this same lines? We were novices in the field, and Sumita came to our, obviously we collaborated with Rajdeep's lab here, but Sumita came to, for the postdoc from the glass research CSIR lab in Kolkata. And what she was able to do was, she helped us make many scaffolds. One was a silicon nanosphere type, silica nanosphere type background or a scaffold. And the other one was graphene oxide based. And I'm not going to talk about this one, but in this case, she was able to make different size spheres, and they were mesoporous holes in it. And depending on the size of the hole, the protein would sit, and you could actually use, you could detect the protein, and we were able to immobilize it really well. But why I'm not talking in detail about this is because we wanted to use it as a device. And with this, we were not very successful in going further because we wanted to couple it to an electrochemical device, so that it's a very miniature device and we can use it. So this, so she also did immobilization on graphene oxide, and this is what I will tell you how we proceeded with making our next stage device. So here again, Shubhankar and Sumita were the 2 main people who were involved in it, and they were able to make this OECT, organic electrochemical transistor based device. And what they did was, so in this device what happens is you have the material is P.PS. And generally what happens is you have an electrolyte, and then you have, this is a negative and a hole nearby it. So you can have a whole current being propagated in this, and so you can have source and a drain, and between them is this material. And so you can have, you can run the device in such a way that the whole current kind of completes the circuit. So you can have, depending on how you have, how much whole current is there, you will have a signal. So what you can do here is, what was done after a lot of efforts, we tried a couple of different strategies, but the strategy that worked best for us was having a gate electrode. This is a gate electrode, having it coated with a graphene oxide and our protein on it. And we did the reaction inside. So every time you do the reaction, phenol binds, ATP is produced, there's change in the overall charge, and that can be detected using the change in this current, and you can then actually detect it. So what went on to happen here was, she was able to first make nice graphene oxide, immobilize it, check that the protein actually is active in these. It does not denature, so this CD spectrum was taken. We were also we also optimized the graphene oxide and the protein so that we can get very nice activity. So couple of concentrations were tried, and again to see if the selectivity remains, that was done. And after that, we were ready with the device, and this is our device, where we were able to see, as you add phenol, you see a drop in current, so it stabilizes, and this is the good part, and then every time you add it again, then it stabilizes. So current is a function of phenol concentration. And that is because when phenol is added, there is a reaction going on inside the cell, right? So every time you can see it stabilizes, and you can keep on detecting phenol to about 2 ppb in this way. So there are certain drawbacks at the end, near the end, but it was still detectable to that particular limit. So then we again tested this with many Pollutants, we also did a lot of environmental samples. Actually, when this chemical science paper came back, we actually had to show, and even for the patent actually, we had to show about 10, 15 different samples, and we did that at different concentrations. Rather, we also got some samples from various polluted sites to show that we are able to detect by 2 different methods this pretty efficiently. So, when we were doing this, the problem that I was trying to tell you a little bit, but I did not focus, was this part. I said we have nice 2 ppb, but the signal to noise is really falling in this limit, right? So, for you and me here, I can say for the paper, we can say LOD is there, it's good. But it is not like we have a very, we are in the limiting region. The other problem with all the systems that I've just told you, every sensor can only do one particular pollutant, right? So 2 problems, we are very close to a very good system, and maybe if we tweak this further, we will be able to get a little bit better signal to noise. The second problem that we were facing was, for each and every pollutant that I have to I have to separately go there and have a sensor A, sensor B, sensor C in different solutions. Can I devise a system where I have very high sensitivity, which will go to drinking water limits? Question A. Question B is, can I detect more than one pollutant at one go, in one solution? These are the 2 questions that we were trying to address here. So one question being, where we want with good signal to noise, good sensitivity. Second question that we had was, can we have multiple, right? So these are the 2 dreams that we started off going ahead. You know, at every stage you have a dream. One time you just want to have a sensor, one time you just want to detect benzene, and when you can detect benzene, you want to add on to make it smaller, then you want to see wherever you hit a roadblock, you try to improve the system. So here we took a very radically, completely changed approach of this whole system. At this point, we decided, let's move into whole cell biosensing, because we realized that many of these systems, there are again some good points for the system, some very bad points. So I'll tell you the good points first, because it helps us. Good points is that you can get very good sensitivity, and because the whole thing is protected by a layer of a membrane, E. coli membrane, you don't have these problems of protein denaturing, this. The bad point is you're using some kind of a genetic material of some kind, but that is contained actually. You never actually leave it in the environment because it's a sensor, right? But you do have that particular thing, and some molecules may not go in that well, but we don't have this problem because remember, in the nature also, these small molecules diffuse inside, and because they're able to diffuse inside so well, that is the reason sensors have been developed naturally for them. So we said go back to the natural system, okay? So what we did was, I'll go a little slow here, and hopefully I'm able to bring across the point. So this system is actually using something called whole cell biosensors. And what I'm doing here is, I'm putting the whole machinery that that bacteria which was evolved to detect phenol inside another bacteria. So I'm using something called synthetic biology here. So the advantage is we can get really nice signal, and I'll show you what we are doing with it. So what is the system? In this system, generally what happens is, so in every system you have a DNA, then there is an enzyme which copies the DNA called RNA polymerase. You will have RNA. RNA is taken into that machine I just showed you, and ribosome, and it becomes protein, right? So if I want to get more signal of anything, because how am I doing the sensing is by getting signal in this case, I need to have more number of RNA. If I have more number of RNA, I may get better protein. So both these RNA polymerase and ribosome should really work well. That's what we want, right? Apart from, so what are the 2 systems that we have here? We have in the natural, the way it works is, this protein, this is a protein. This protein is a sensor protein. Remember I showed you the one which has a sensor domain, the ATPase domain, etc., etc. And in the cell, the minute we put it, the minute it sees this particular, as you can call it a cassette, this genetic circuit, it will, this PM thing is written, is an RNA polymerase type which sits in something called a promoter. So it will copy this whole thing, and that RNA will then make protein. So this is done automatically in the cell. The minute I expose the cell to this particular cassette, this will start making a lot of this protein. But this protein is only made when the phenol comes. So this protein will detect phenol. When this protein will sit on this PMOP, whatever that is, this is just a promoter, then you will see another RNA polymerase will come and make all these degradation genes. So I don't know whether I'm able to explain it. Just wanted to tell you is there is one protein which is automatically being made. The second system is made only when the pollutant comes. The first protein that is automatically being made senses the pollutant. When it has enough pollutant, then it starts the RNA of this second set. So obviously we cannot make all these proteins. So what we did was we took the same machinery and we put some kind of a reporter gene instead of all those. So now what is happening is every time there is phenol in the system, some kind of fluorescence is coming, right? And we are detecting that because this protein is GFP, for example. That's what we did. So you can see here, what we did was we wanted to do multiple detection at the same time. So what we did was we changed this reporter by making the mutations. So GFP is nothing but a protein which looks like this barrel and has this, this is the fluorophore. There is a dehydration reaction which happens between serine, glycine and a tyrosine, and this kind of a compound is made which is fluorescent. And you can change the properties of this compound by changing it into a tryptophan or a histidine or keeping something close by which will kind of change its properties. So as a result, you can have a fluorescence which is GFP-like or yellow colored or blue or cyan. So what it is doing, why am I doing this? I am trying to make a system which has different reporters. Why do I want that? Because one will be for phenol, one will be for benzene, one will be for toluene, one will be for xylanol. So what will happen is because my sensor is different, then what am I doing is I am going to get one sensor system, one color, another sensor, another color, another third sensor, another color. So I am making a palette of colors depending on the sensors. So I can basically stitch. I can stitch my engineered protein and whichever sensor I want. So I've made a phenol sensor, a xylanol sensor, which is a benzene sensor and an ethyl benzene sensor. And they all have slightly different spectral maxima. So you can, although there's an overlap, so you can use this to be able to detect multiple pollutants together. So Rohita was a joint student with Arindam in IIT. Worked on this project, started working on this project, and what we were able to show was that we made the sensor, but we ran into some problems. The sensor had, we wanted to do 1 ppb, remember? And now we have an LOD of 100 ppb. So we have gone back, right? So then she came and she said, oh, it looks beautiful. I'll just show you the pictures of it because we were doing a lot of imaging with this. I'll show you the images. So you can see, these are all the E. coli cells. And you can see green color, blue color. I mean, these are false colors, but you know, in the spectra you can see if you try to overlay, but they have different spectra. So you can detect these molecules and everything is good. But where have we failed? We have gone back to 100 ppb. So, Shamita was who started the project. Rohita and I got really bugged with this thing. I said, oh my God, we were going to make a better sensor. We ended up with a worse sensor. But one thing that we were able to do was get all the colors. So then Rohita and I thought about the problem a little bit more. So we did screen at different times, and we were able to make a standard curve for this. But we thought about the problem a bit more, and we said, what is the problem? How do we improve the sensor? The problem is GFP by itself. GFP is not a very good fluorophore actually. We can get better with luciferase, but then you are not seeing anything. You know, it's not in the visible range, right? So yes, we could change the reporter, make it a little bit better reporter, but can we do something else? So we said, this is something that we can do. This is our problem. I can try to find something which will help me make more RNA. Then if I have more RNA, I will have more protein. I can also make this protein making system a little bit more efficient. Then also my signal will go up because my sensors are now just, they're on overdrive. You can put it that way. And I don't want it for long. Even if the cell works in overdrive for half an hour, that's my detection time anyway. I don't care. After that, my sensing is done. So what we did was, we decided to look at the genetic circuitry which will help me make more RNA. Right? So there are many promoters which are already present that you can tweak. And that what it does is, this is called promoter strength. What it pretty much means is a more comfortable seat for RNA polymerase to be there. It is more welcome. If you have a certain DNA, the structure of the DNA is slightly different, and as a result, RNA polymerase is able to come and feel happier there. As a result, it makes more RNA. So what we are doing is increasing the total number of RNA. If there is more RNA, more protein will be made. So we screened through some of these things, and you can see that for one of these promoters, you can see very nice enhancement. Right? Then we said, we have made this good. Why don't we make the ribosome also happy? So every time a ribosome wants to work, there's something called a ribosome binding site, RBS. We said, let's also make the ribosome happy. So we have made RNA polymerase happy, so we have more RNA. We made a ribosome happy, so we have more ribosome working more efficiently, right? And as a result, what is happening now is, with doing this, we are able to increase the dynamic range. So you can see this is what the sensor was working like before. And now, can you see the sensor has gone up. The overall signal has gone up. We have a good range. Now we are able to get no longer as flat in the region that we wanted. Our signal to noise, the overall signal is itself been enhanced. And now when we go, we are able to see in the 1 ppb or even sub 1 ppb range, very nice signals. So what has happened is, we had a roadblock, and we just used a little bit of help from our synthetic biology community, and we were able to kind of circumvent it. What we were able to then do is, is that we were able to quantify this data with a lot of care. And what we were able to do, you can see these lines are there, which is trying to use the maximum signal which is available at any point, and then trying to see if we can benchmark the signal. And so we are doing thresholding, which is minimal thresholding and maximum thresholding. And we are taking a statistical average of the signal for each concentration. And we are coming up with points here, so that we are able to quantify the signal. And we have seen that if we take an unknown sample and we take two methods, we are able to then use this. So using this method, we are actually able to quantify now very low ppb, if we would like, in the cells. And and the good part is, it's more visual also, right? And not only that, I'm very far from the source, I guess. Not only that, what we are able to do is, we are able to show that we do not have interference. So this is, I'm adding more than one sensor, but I'm only having phenol, right? So it doesn't have any cross. If you add other molecules also, it doesn't show any signal. Or if I have this particular sensor that I've engineered, I add other molecules and it doesn't show. So you can see that they're pretty specific sensors and standalone. And then you can combine them together. When you combine, that means you can put more than one sensor together. And in this case, what we do is, we do spectral imaging now. Don't just look at the color, because it's a maxima, so now when we combine cells, you can have a cell which is cyan, you can have a cell which is blue, and we have a cell which is yellowish and greenish, false colored. But the spectra is much more reliable. So what we do is something called spectral imaging, where we go, and this is the work with Arindam that we have done in his lab using the epifluorescent setup. You can see here, we have two pollutants here, and then we can pick up cells and we can show exactly that it is not like we are dreaming this color is there or not. We are able to see by spectra. There are overlaps and there are problems. It's not like full of, but you can see this is a two different pollutant system. This is 3 different pollutant system, where you can again see, you can choose a cell and you can tell, and then you can do statistics after that. And you can have a 4 pollutant system also, and so on, where this is still 3. where you can just show that you can detect these pollutants, something like this. You know, you can have a water sample where you have all these, you can pick out your cells and you can then do your averaging based on. So it could be that their levels are not same. Maybe benzene is more, phenol is not there that much, so you'll have number of cells that you add of each pollutant type is same, and then you may end up depending on what you are trying to look at. So you can, basically what you can do now is, you can have multiple pollutants, you can bypass the problem of limit of detection by using some genetic circuitry, and you can come up with this whole thing in a much better format than we started off with. So, so when we did this, we thought that's really great, and as always we were also very worried about the shelf life. So what we did was, we realized that what we can do is, we can freeze dry our cells, add water to them or little bit of media, and you can actually bring them back. Sorry, you can bring them back and they still work the same. And you can do it for 6 months at a time. You can look at different low and high concentrations, and these are the sensors. And this is just one of the experiments after a month. But you can keep doing this, and now you have a sensor which has a in a tube, a freeze-dried thing, and it just stays. So you have your whatever system of sensor that you have thought about is in a small tube. It never gets out of it, and you can do the whole reaction within half an hour, and you can get your colors. Right? So this is what we were doing, and we wanted to still keep on making, because the things that I was trying to show you still had Arindam's lasers being used, because we were doing proof of principle. But we wanted to come up with a cheaper device, which we have not achieved so far, but we are in the works to do that. And what we did was, we thought about, at least we were just thinking, Manu Prabhakar in Stanford has a very nice folderscope, which is for just not fluorescent. Now he's come up with one, but we still don't have it. But what we were trying to do in our lab was, can we modify this so that we are able to get initial results where we are able to see phenol, xanol, benzene, etc. And we are modifying this setup. It's not done yet. It's in its trial and error phase right now, where we are trying to look at this folderscope and see if we can actually modify it so that we can have at least the first check of our sample, even if it's at higher concentrations. Before you bring it to lab, you can check whether that water is heavily contaminated, not so contaminated at the site itself. For our other portable things that we had, they are actually can work on site, like our electrochemical sensor and this. Maybe we can use both in conjunction, actually. That is also a possibility. So, so with this, I just want to tell you two things, which are just two slides. I don't know whether I've gone above my time. I never looked at it the time. Huh? Yeah, because I'm just also talking very fast. I have this problem. But this, we are going to PCB. I told you we are in mono aromatics. We are going to biphenyls now. And we want to do PCBs and other biphenyl sensors, which are, one of them is also, it was suggested to me that for plastic, the degradation product is a bis biphenyl. That we can also now use directed thinking to evolve for it. But we have till now only looked at this particular problem, and we started with a sensor, natural sensor system, which looks like this. It is for hydroxy biphenyl, and we did a lot of tweaking with the sensor, and right now we can do some preliminary things where we can do chlorine and a methyl. We are going towards a PCB type of sensor. So we are evolving it currently. Apart from that, we are also looking at some basic work with Jagannath Mandal's lab at TIFR, where we are trying to understand more than just this problem, where the problem is, how does this protein actually finds phenol? Is it just the binding site or the entry pathway? You know, it's like if you want to enter this room, there's a whole entry pathway. If I put blocks in this entry pathway, then it will be a different pathway, because this pathway and the stairs is a different pathway, right? And so maybe this is I don't know, this is actually easier with gravity. But what I meant was there's a distance difference here and a distance difference here, height difference here and here. So what is happening is the entry pathway is very important, because ultimately somebody has to reach me, right? Either from there or here. And what are the access points and how they can be blocked. So in proteins also, to make sure random molecules don't go and bind, there are entry pathways. And we figured out that this entry pathway is pretty complicated. It rejects benzene or other molecules. And for acceptance, how do you tune that? Because you don't only have to tune the active site, you may also have to tune the entry pathway. And we did a lot of work where we blocked the entry pathway and showed that the molecule cannot reach. But I'm not going to talk about it because it's already been a long talk, but pretty much some basic work in that direction is also going on. With this, I'm going to stop, and I'll just say that we started with a simple sensor where we tried to figure out how we can use natural bacteria to sense phenol. And over the years, we have had many works. Some of the works are more basic, where we try to understand allostery of this protein. We're actually also doing cryo-electron microscopy of this protein, since we are structural biologists. And we are using different kinds of, in collaboration, ways to go ahead and see if we can actually reach to a step where we can use this as a sensor easily, right? So that was my journey and all the journey of my students. These are some of the people who participated. Shamita, I told you, most of them are actually, Shamita has already gone out to Harvard. He's just left for Imperial College. She's in Tulane and she's going to Stanford. So all these students have graduated and we have a new batch coming up. But this is an old picture of my lab where we work together. And without the students, without each other, we cannot really survive, because we have ideas from each other, and most of the work is now carried on by graduate students. So I am very thankful, and I love my lab. I love the work we are doing, and it's an exciting thing to be able to come up with a problem and then nudge your way through it. So that's what I wanted to spread the message, and I'll be happy to take any questions. I'm sure I was like a race. I was in a race. So thank you, everyone, and open to questions. Thank you.
One question I have is here.
Yeah.
So you're talking about detection.
Yes.
The next step is purification of that, right?
Yes.
Is there already some ideas about that, or?
So we started doing that a bit, but right now we got so bogged down with the two things our lab is doing. We got into detection, and we also have a huge effort on antibiotic resistance that I didn't talk about. But what we did was, initially we took these bacteria, enriched them on a carbon scaffold, and we made these discs, and we tried to purify water. And we were somewhat successful, but then we did not, it's just that we have not done it. We have not pursued it because of lack of, time, I would say, on my side, but it's going to be challenging. It's not that easy.
But in normal purifiers like AquaGuard or something like that, does this go through?
This will, if it's a bacterial this system, you cannot put it in an AquaGuard for sure. But the electrochemical sensor, maybe because the protein is very stable for a very long time, because it's 4 months, 5 months, we have seen it's possible. But nobody is going to accept a bacteria in their AquaGuard.
No, my question is, in a normal AquaGuard kind of a purifier,
they don't remove these pollutants at all.
Oh my God.
None of them. Because benzene, they're in the low levels that are there, they're low levels, and they're carcinogenic in very low levels. Yes, yeah.
Yeah, so you talked about the sensitivity and optimizing sensitivity using promoter modification and all. That's all very well. But you said something about, half an hour incubation to see the, expression and color of the fluorescence and all. but if you do increase time, because once the phenol is there in the system, it's going to be
Yeah,
constitutively expressing.
Uh,
So can you just with the extended time increase sensitivity also?
Yes, we can we can extend the time, but the problem is when you extend the time, we have no problem. The signal just keeps going on, but nobody wants to wait in this world for like say 4 hours later you can see your signal. So we were trying to do something where you don't see it, and also every protein in the cell has a life cycle, like there's a time, because it's inside the cell. They will also be taken away for degradation. So it's a between degradation and production there is a balance. So and the time you want to use for detection. So yes, you are absolutely correct. We have gone to higher times and we can get better signal, but we tried to do something where we don't have to wait for a very long time.
So the one more question is, you talked about using microscopy to detect. So that can be a limitation for application. So what are the other ideas for detecting the light without microscope? Can you use some kind of a light sensor for that?
You can just use a fluorimeter. You can always detect. People just love looking at. So looking at you know, you can see when you can, if you have a fluorescent setup you can, but if you are going to use a fluorimeter, the problem is you will not be able to tell one cell from another, right? So you cannot do multiple detections at the same time, but you can do detection. So I can have A pollutant, B pollutant, C pollutant, D pollutant separately, and then I can just use a fluorimeter. But if they are mixed together, then it becomes a little more complicated because you cannot tell A from B. Multi-pollution is a problem, otherwise not.
In interest of time, we'll have 2 more questions, one by Sayan and the other, there's a student at the back.
I would like more students to ask me questions, you know, I
Yes.
would encourage it even if it's possible.
The student can ask first.
Yeah, sure.
Student, does she have a mic?
Don't feel inhibited. It's okay. I also don't know much. Yeah, sure. Whatever makes everyone comfortable. No, you can ask if.
Very nice presentation. I have one basic question. Currently we are facing a microplastic pollution. So in that aspect, whether the oligomer detection is easier with the such biosensors, or only the monomers are the one which can be recognized?
See, I don't have an answer for this because we have to do a little bit of research to see if we can find a system which detects some kind of a polymeric unit, right? You can start with a system, and you know, when I was showing by eye, we were doing it. Maybe that's not the best way to do it. You can do something called directed evolution, but you have to start with a close enough system, right? So if you find a close enough system, close enough doesn't mean exactly same, but something that can take a polymeric chain of some kind, and some structures are similar, you can then perhaps use that, right? There are those fatty acids ones which have a very long chain thing. So depending on that, but then you'll have to do directed evolution on that system. But the monomer one is easier to do because we have an enzyme with us which is very close, it's already doing biphenyl. So I can actually tweak it faster. I can do by trying to do intelligent design, or even I can use this system for directed evolution and come up. So if you are closer to the solution, the thing comes faster. But if you start very far from the, you know, to come there would be difficult. So if you can find, if we or whoever is working on it finds a system close, and then I have a feeling they'll have to do directed evolution for this problem, for the polymer.
Okay, not even with the oligomers.
It will be difficult. It's, you can, maybe small oligomers, but you'll have to really find a system suitable for it if you want to do it. I think one should not go the enzyme way there. Everything doesn't have to be the enzyme way, right? You can, enzyme because it's working. Otherwise, think outside the box. But for monomer, maybe the enzyme may work.
Thank you.
So 2 quick questions, one from Sayan and one from Parveen, and then we'll close this session.
So, Ruchi, nice
Yeah.
talk. I have 2 questions. The first thing is from the first part, where you initially had the phenol, which was forming a hydrogen bond between a tyrosine and a histidine. No, not tyrosine.
It is tryptophan
And
and
a
histidine.
Right. So the thing is, when you change that something, whatever,
changed it to a tyrosine.
And then you have a
histidine into
benzene
tyrosine.
derivative that actually binds there, right? A benzene derivative now binds there instead of
Benzene binds.
What interaction is driving the different benzene derivatives to bind in that pocket?
So most of them are hydrophobic. So I'll answer your question in a long way. Since Jagannath and I have spent a lot of time on it, we figured out that the binding is not just the binding pocket. There is an entry that happens, and there is kind of a dynamic tunnel. And there are a lot of hydrophobic amino acids at that, which kind of squeeze in the hydrophobic part in. So what happens is there is part 1, it gets in, then it moves in the middle, and then it goes to the binding pocket. It is not like this, like that it gets bound, okay? And that's where we try to tweak those residues and show that the molecule is not even detected if you change that path. So it is not just the binding pocket. The binding pocket is the end result where it's comfortable, because the rest of the residues of the binding pocket are very hydrophobic, and it's just a nice environment where you have isoleucine, some phenylalanine, some kind of a padding around it, so that it is in the right solvent, let's put it. Okay. Benzene is not with water, but it is with its wherever it wants to be.
So the second question is, see, when we were talking about this different phenol and the benzene derivatives and we're detecting in the last part, not all the derivatives have the same sensitivity in the detection.
Yes, yes.
So when you talk about the detection limit, like 0.1 ppm, ppb, what you were talking about, this is for the ones which are least sensitive or not?
So for the when we do the protein level, we have huge, not huge, but there is some difference in sensitivity because it's a natural phenol sensor. But when we are doing the cells, we have such a lot of signal to noise, it does not matter. We are able to get below 1 ppb for all. Okay. I mean, we can get sub ppb for some, maybe phenol, but we don't care because for drinking water the limit is below 5. Once we have reached there and we are already in 1 ppb, I mean, what is the purpose of increasing sensitivity? So okay. we did not worry about it.
Okay, thank you.
Yeah.
We'll open the floor to a question from a student. Any student wants to?
Ma'am, if phenol concentration is very low, then PMOF, how to detect less amounts of phenol?
Yes, it's in ppb.
ppb.
Very, very low.
Yes,
nanomolar.
how to detect?
Low nanomolar actually. That's where you are. So you are already in very low limit. See, even in benzene, if it was in high limit, you will actually not have any solubility in water at all. It is a problem when if it's very large, you will see something in your water, right? You will already say, I'm not going to drink this water. But when you don't see, and it is there is where the problem lies. That means in the low solubility limits when you reach, phenol is not a problem, but benzene, toluene is a problem. That's the limit that we are worried about. Many people also ask me about high detection limits. High detection limits are not that much of a concern. You can more easily detect that because there is a phase separation. It's the low limits that we are more worried about.
It was a very interesting talk. I like the way how your research changed as each student came in and it got evolved in that time. So my question is, if we use the bacterial sensor, then what is the quantity of water it can detect in one batch for a bacterial sensor?
So we use very little water at one point. It's a few microliters, but you can have a big batch and you just need enough to detect in that. You don't need to, I mean, it's a solution, right? So very little water, maybe 1 or 2 microliters is enough.
If we try injecting it in a bigger system or...
So if it's a solvent, you're assuming that if it's a batch of water, it will have the same solubility. But you can always, if it's a river or a big thing, then you can have sites and you can cover areas and then figure out if there is more pollution at this one end as compared. There can be not polythene in this case. Suppose there was an oil spill which was long time ago, or there are some packaging materials which were there and then they slowly release many of these chemicals like xylanols, for example. So there you'll have to do sites. So yeah, depends on the what you are trying to monitor. Right.
Thank you.
With that, let's thank Professor Ruchi Anand once again for this taking us through this wonderful journey. of biosensors. And before you move off stage, and I think you need to stay on stage.
Okay.
I'd like
Do I
to-
need a volume?
No, maybe not. So I'd like to invite Dr. Premnath to felicitate you.
Thank you so much.
So I'd request both of you to stick on here on the dais. The next part of our event today is going to be conducted by students. Subhashri and Rajlakshmi are going to host that part of the event. So I invite them to take over and start the award distribution.
Thank you, ma'am, and a very good morning to one and all. You can come this. You can come here. You can. Audible, right?
Yeah, a very good morning to everyone present here.
So the journey of a PhD is a long one, and it is a very challenging work. So while we do it for intellectual stimulation, sometimes we wonder, why do we keep ourselves and put ourselves through such difficulties? We tend to revise a single chapter countless times before sending it to our supervisors for feedback because we think that it might not be right. But the problem is the right manuscript or the right chapter does not exist. So it is an illusion which painfully gets shattered while we get back critical comments from our supervisor. While on the other hand, a piece of work that we might think is just an average could be good enough for our supervisor. So it's good to have high standards, but never to confuse progress with perfection. So the journey of PhD is a long one, where it is very easy and normal to feel demotivated for a stretch of time. So we all need a little bit of inspiration to perk us up during those days. So today is one of such days where the entire NCL fraternity has gathered together to acknowledge and to celebrate the tedious hours of fabulous and dedicated research that is done by our fellow researchers. I would like to here extend our acknowledgement and gratitude to the NCL Research Foundation for recognizing as well as rewarding the scintillating work that is done by the students.
NCL Research Foundation is a non-governmental body which was registered in 1990 as a non-profit public trust. The primary aim of the foundation is to motivate and recognize the high level of team and individual efforts of scientists, staff, and research students for their contribution to sustaining the values at CSIR-NCL, namely sustained excellence in science and technology, contribution to the creative wealth in the society through science and technology, individual efforts beyond the call of normal duty, and constant endeavor to improve productivity and efficiency. Through several named lectures, NCL-RF also honors the men and women who made NCL proud and in this way keeps the legacy alive, paying tributes to the lasting contributions. This organization is fully supported by the donations made by individuals and companies and is managed by a distinguished board of trustees drawn from academia, industry, and public life. The NCL Research Foundation appointed an internal committee that played a pivotal role in selecting the award winners. We would like to extend our gratitude for their efforts in doing the same.
So with this brief introduction, I would like to move on to the announcement of the NCL-RF Awards for the year 2023. And as we have with us Dr. Premnath Venugopal and Dr. Ruchi Anand to felicitate the awardees. So the following awards will carry a certificate of commendation and a cash prize. It is to announce that the cash prize will be given to the awardees in a later half, which will be intimated to them. So moving forward with the first category. The first category of the NCL-RF Awards is the Best Research Scholar for 2023. The first award is Mrs. Uma Devagupta and Professor Ashwini Kumar Nangia Award for Best Women Research Scholar in Interdisciplinary Research. And the winner is Ms. Kranti Salgaonkar. The next award is NCL-RF Kirti Sangoram Memorial Endowment Award for Best Research Scholar in the area of Physical Chemistry, Material Science, and the award is shared by two. First, Mr. Siddharth Barik and Mr. Siv Murthy BP. Come fast, come fast, come fast. Siddharth then Siv Murthy. Next is for Chemical Sciences, and the award is shared by Mr. Anand Sabde and Mr. Viksit Kumar. Anand Sabde. Mr. Viksit Kumar. The next award is for Catalysis Division, which goes to Mr. Rohit Kumar. We have two award winners for Engineering Sciences, Mr. Muzammil Sarfaraz Khan and Ms. Rajashri Jundale.
In the second category of awards, the second category of awards is for the best research papers in 2023. The first award in this category is the Dr. Rajappa Award for the best published research paper in organic chemistry with highest impact factor. And this has been published in the journal Green Chemistry. The award is bestowed upon Chandani Pradhan, and one of her friends will collect it on her behalf. A loud round of applause to Chandani Pradhan. The next award is the NCL-RF Nanai Natu Award to research scholars and the project assistants for the best published research paper in organic chemistry with the second highest impact factor. And this has been published in the journal Organic Chemistry Frontiers. This will be awarded to Sairam Dhyaneshwar Veer, who is also not here. So one of his friends will be collecting it on his behalf. A very loud of applause to Sairam. The next award is the NCL Arif Gupta Pardeshi Saini Award for the best research paper in biological sciences with highest impact factor for the year 2023. And this has been published in the Journal for Cleaner Production. And it has been awarded to Pranav Naya. A loud round of applause to Pranav Naya. The next award is Dr. Krishnan Award for the best published research paper in material sciences. And this has been published in the journal Angwante Chemi International Edition. And the award winner is Viksit Kumar. A loud round of applause to him. The next award is the Dr. Krishnan Award for the best published research paper in computational sciences. And this has been published in the International Journal of Hydrogen Energy. And the award winner is Ashwini Verma. A loud round of applause to her.
The next award is Dr. Joshi, Dr. Shivram Award for Best Published Research Paper in Polymer Science and Organic-Inorganic Hybrid Materials for the journal Agudatic Chemi International Edition goes to Mr. Sairam Veer. I would like to request someone to collect it on his behalf. The next is Dr. B.D. Kulkarni Award for Best Published Research Paper in Chemical Engineering Technology. And the journal is Chemical Engineering Journal, and the award goes to Mr. Ravi Ranjan. Please give him a big round of applause. People are not coming from that way, everybody is coming from this way. The next award is Dr. Paul Ratnasamy and Dr. Ms. Sunita B. Kulkarni Award for the best published research paper in catalysis for the Journal of Material Chemistry A goes to Ms. Kranti Salgaonkar. The next award is Dr. Patel Joy Award for Best Published Research Paper in Physics for the Journal of Chemical Physics goes to Mr. Srijan Chatterjee. I would like to request someone to collect it on his behalf.
The next is the Dr. Saurav Pal Award for the best published research paper in physical chemistry or chemical physics. And this has been awarded to Siddharth Barak. This work is featured in the journal Small. the Dr. Patail Joy Award for the best published research paper in physics goes to Palak Patel, and her work is featured in the Journal of Chemical Physics. One of her friends will be collecting it on her behalf. Next, we have the much-awaited award, which is the Best PhD Thesis Award for the year 2023, and this is shared between two people. The first is by Dr. Apurva Patrake, who worked with Dr. Manjusha Shelke, and the second recipient of this award is Dr. Siddeshwar Ankade, who worked with Dr. Benudar Punji. Let's give a loud round of applause to the both of them.
Hello, thank you NCL for the award. I would like to receive this along with my ma'am. Ma'am, please come on stage. It is possible. Yeah, it's thesis.
He will be collecting it on behalf of the next candidate.
So next up, we have the award winners for the poster presentations. So first is from Organic Division, Organic Chemistry Division. It is shared by 4 people. So the first award goes to Megha Palange. The next is Sayali Jagtab. The next best poster goes to Sarad Suryavanshi. The next award for best poster from Organic Division goes to Ganesh Padi. Where is Ganesh?
Up next, we have the winners from the Chemical Engineering and Processing Division. The first award goes to M. Anirudh. I request him to come forward to collect his award. Or if one of his friends or lab mates could collect it on his behalf. The next award winner is Muzammil Anwar Sarfraz Khan. Let's give him a loud round of applause once again. Next, we have the winners from the Polymer Science and Engineering Division. Next, we have the winner from the Polymer Science and Engineering Division, and we have Radha Kishan Sahadev Kargude as the winner. But I request one of his friends to collect the award on his behalf. Yeah. Yeah.
The next is for the posters presented in Hindi language. And the best poster award in this category goes to Arindam Saha. It is also shared by Ms. Pratiksha Pandey. Next up, we have Best Poster Award is from Biochemical Division, and the first prize goes to Amreen Sheikh. If she is not around, can anybody collect it? Okay, we'll move on to the next. Best Poster goes to Manali Bajpayee. It is also shared by Sai Prashant Joshi. Is he around?
Next, we have the winners from the Physical and the Materials Chemistry Division. Firstly, we have Mayank Yu Singh. The next one is Atira Naya. And the third winner is Arindam Biswas. The winners are Mayank Yu Singh, Atira Anaya, and Arindam Bishwas. Finally, from the Catalysis and Inorganic Chemistry Division, we have the winners as Pooja, Amrita PR, Akash Bhatkar, and Shambhaji Shivaji Gadge. I invite Amrita PR to collect her award. I invite Akash Bhatkar. And finally, Shambhaji. Okay, one of his friends is collecting it on his behalf. With this, we come to the end of the award ceremony. I now invite Dr. Anurag. Here's an announcement for the RF Award winners. They can collect the cash prizes, checks from the TMG office on the 5th of March, which is Tuesday. Thank you. With this, I call upon Dr. Raghunathan for the vote of thanks.
Thanks, Premnath and Ruchi, for doing the award ceremony. Hello. I would request people to stick on for at least 2 minutes. 120 seconds is not much. Hello. Since I've been co-opted to deliver the vote of thanks, I'll try to make it really brief. On behalf of entire NCL, I would like to express our sincere gratitude to the incredible event management team for making today's National Science Day event a resounding success. I thank the entire event management team that consists of an exhaustive list of people whom I will not name here, who have been involved right from the initial planning stages to the final execution of the event. Their dedication, creativity, and meticulous attention to detail are truly evident in ensuring a smooth and seamless experience for everyone involved. As we draw an end to the National Science Day celebrations, I would like to remind us all here of Stephen Hawking's quote, "Science is not only a discipline of reason, but also one of romance and passion." So, let your new year in science begin. Thank you.